So here you can see in an agar plate we can see a female laying eggs. Eggs are very small in size, and if we look at them at the electron microscope, you can see here we have the anterior side, the posterior side, and dorsally we have one pair of respiratory appendage at this stage, and these respiratory appendages have important roles in respiration, since the trachea, the main respiratory organ, is not yet formed in this Drosophila egg. So in Drosophila egg, the body plan is already organized. So even before the fertilization, the Drosophila egg has its anterior pole and the posterior pole established. So it means the anterior posterior axis of the Drosophila egg is established early, well before the fertilization of this oocyte. And this is due to the accumulation of specific maternal messenger RNAs like bicoid messenger RNA in the anterior cytoplasm, nanos and OSCAR, both together they will be accumulated at the posterior cytoplasm. These messenger RNAs, they are very important, and their expression, their maternal expression early is important for the establishment of the anterior posterior body axis that will be maintained for the whole Drosophila life. So the micropyle, as I mentioned before, it's the site of sperm entry during the fertilization of the Drosophila egg. This micropyle is located at the dorsal, this is the dorsal side, this is the ventral side, this is the anterior, the posterior. So the micropyle is located at the dorsal anterior side of the Drosophila embryo, where the sperm will penetrate. So the micropyle is a very small tunnel present in the chorion, which is the outer egg shell of surrounding embryo, and this micropyle is probably preventing the polyspermy. It means it will prevent any penetration of additional sperm cells once one sperm cell entered and fertilized the oocyte. So in this case, it's different from what we have seen, for example, in amphibians and mammals and sea urchins. So we don't have any cortical granules to block the polyspermy. So we think that the micropyle is the structure that will block the polyspermy at this stage. So after the fertilization, in Drosophila, we have different rounds of cleavages to form the blastoderm, okay, or which is similar to the blastula in other organisms, okay. So we call it blastoderm in Drosophila. But how the blastoderm is formed? So we have different rounds of nuclear division and not cellular or cytoplasmic divisions, okay. So as you can see, only the nucleus will be divided, okay. We have nuclear division, so this nucleus in the fertilized egg will be divided to give 2, 3, 2, 4, 8, 16, etc. nuclei, okay. And these nuclei will be accumulated in the center, okay. So in the central part of the cytoplasm, it means right here, okay. So these numbers from 1 to 14 refers to the number of cycles, okay, to the number of cleavage of the nuclear cleavage cycles. So from the cycle 1 to the cycle, let's say 9, we have lots of nuclear divisions, and starting the cycle 10, okay, the cycle 10 to the cycle 13, we start having a kind of nuclear migration from the center to the periphery, so from the center towards the peripheral side of the embryo, okay. and by the cycle 13 we will get a syncytial, okay, blastoderm, okay, syncytial blastoderm, when so all the nuclei have been migrating to the periphery. Okay, so these stages define the syncytial blastoderm stage, while the cycle 14 define the cellular blastoderm stage. The cellular blastoderm, it's when the nuclei, okay, are located at the periphery. Each nucleus will be surrounded by its own cell membrane, okay. And as you can see here, so of course cycle 14 is called the cellular blastoderm stage, since all the nuclei will be surrounded by cell membranes. We have a cellularization of the cells, okay. But if you look here, we have groups of cells that have been segregated from the other nuclei and surrounded by specific cell membranes. These are the pole cells. They are called pole cells, and these pole cells located posteriorly, this is the anterior side, the posterior side, these pole cells are located posteriorly, and those are germ cells precursors. Okay, they are the precursors of all germ cells and embryos. So here you can see the nuclei are stained in these micrographs, okay, taken by a laser confocal microscope, okay. And I remind you that the cleavage in Drosophila is superficial, okay, so the nuclei will be cleaved in the center of the embryo. So as we mentioned before, the cycles 10 to 13 define the syncytial blastoderm. Syncytial, it means when inside the same cell you have many nuclei, and these nuclei are not surrounded by cell membranes. Okay, these different nuclei are present in one single cell. This form is called syncytial, okay, and this stage is called syncytial blastoderm stage in Drosophila. Okay, so you can see here the syncytial blastoderm stage, when all the nuclei have been migrated from the center to the periphery, okay, and once these nuclei are completely cellularized, so each of them is completely surrounded by its own cell membrane, the stage is called cellular blastoderm. Okay, this happens at the cell, or this defines the cell cycle 14, okay, called stage 5 here in this figure, okay. And usually 2 hours and a half to 3 hours maximum after fertilization we reach the cellular blastoderm stage. So the cell cycle 10, okay, the cell cycle 10, or the stage 3 right here, marks the first signs of transcription from the zygotic genomes. So few zygotic genes start to be expressed by the cycle 10, okay, only few zygotic genes, but all other genes are still maternal, okay. So here this diagram shows us the early development in Drosophila from the fertilization to the cellular blastoderm stage. So after fertilization we have a fusion of the sperm nucleus and the egg nucleus to form a diploid nucleus inside the cell. This nucleus, okay, is fusing 30 minutes after fertilization. Okay, these nuclei are fused 30 minutes after fertilization. So 70 minutes after, okay, we start seeing many nuclei. So here this phase is a phase of rapid nuclear divisions, okay. So we have like one division every 8 minutes, okay. So that's why 70 minutes after fertilization we already had 9 divisions, okay. 9 times 8 equal around 72 okay, 72 minutes. So by the cycle 10, and from the cycle 10 to the cycle 13, okay, we have also nuclear divisions, but we start seeing nuclear migration from the center to the periphery, okay, and 2 hours after fertilization we have this syncytial blastoderm. So the cycle 13, or the syncytial blastoderm stage, is characterized by a slow nuclear division. So now we have one division every 25 minutes, okay? So it's quite slow, this nuclear division, compared with this stage, okay, with the beginning of syncytium formation. And finally, by the cycle 14, the nuclear divisions become asynchronous. They don't happen all of them at the same time. And by this stage, so all the nuclei, okay, like 30 hours after fertilization, will be surrounded by cell membranes. And this defines the cellular blastoderm stage, or the cycle 14, which is asynchronous. So some groups of cells complete this cycle a bit earlier than others, okay, but like 3 hours after fertilization, all the nuclei will be surrounded by their own cell membranes. So here we have two micrographs taken under a fluorescent microscope. The first one is taken from a syncytial blastoderm stage, so it means the cleavage cycle 10 to 13, while the second figure here is taken from a Drosophila embryo at the cellular blastoderm stage by the cleavage cycle 14. So it's easy to recognize that this stage is syncytial blastoderm and this one is cellular blastoderm. So here we see only the nuclei, okay? We can see only nuclei, while here the nuclei are surrounded by cell membranes, since they have done a staining, immunostaining here, and with actin, which is a main component of the cell membranes. So we can see that these nuclei, all of them, are surrounded by cell membranes. So it means the embryo nuclei have been cellularized.